a la última conferencia, una conferencia, creo que diría yo que inspiracional de uno de los líderes, de las autoridades mundiales en genética y diagnóstico prenatal, el doctor Denis Law, quien fue el que descubrió o fue que inventó el DNA fetal en sangre materna. El doctor Oscar ha presentado un mini currículum que realmente cuando ustedes revisan el currículum del doctor es impresionante, y él muy amablemente lo contactamos y se prestó muy amable para presentarnos esta conferencia. Oscar, adelante.
Sí, en una diapositiva no caben ni las fotos que tiene con todo el planeta, ni los premios que se ha ganado, ni todos los estudios que tiene. Pero Denis Law nació en Hong Kong en el año 63, en la época en que Hong Kong era una colonia británica. Recuerden que Hong Kong perteneció al Imperio Británico más o menos 150 años. Entonces, de pronto esto le facilitaba a todos los chinos de Hong Kong ir a estudiar a Inglaterra. Él estudió primero en Cambridge, un preclínico, después en Oxford hizo el entrenamiento clínico con su PhD, es profesor de patología química en la Universidad China de Hong Kong, director del Instituto de Ciencias de la Salud de Li Ka-shing, patólogo químico con interés en patología y diagnóstico molecular. Tiene más de 290 publicaciones ya siendo autor o coautor de estas, y él desde 1989 más o menos empezó a ver en los estudios de DNA de células cancerosas que circulaban en la sangre, que se interrogó si de pronto las células fetales, el DNA fetal, podría estar circulando en sangre materna, y más o menos en el año 97 publica en la revista Lancet la primera publicación, perdón el pleonasmo, de su estudio, en donde demuestra la presencia de un porcentaje importante de DNA fetal en sangre materna.
Pueden oír la conferencia en inglés o en español, la traducción. Moviendo el Zoom. Adelante con el doctor Denis Law.
I'd like to thank Professor Jose Enrique Sanden-Blair for the kind invitation. It's my honor and privilege to give a talk today to talk about my journey in inventing non-invasive prenatal testing and some of the future development in that field. So this is a picture of myself taken over 30 years ago when I was still a medical student at Oxford. So as a medical student, I have to study different subjects, and one of those was obstetrics and gynecology. And I learned about practice of prenatal testing, which at that time, if we were trying to test the cells or DNA from a fetus, we have to insert a needle to do amniocentesis. And of course, this process is associated with certain risks. So as a young medical student, I was thinking, is it possible to develop a safer method just by taking a blood sample from a mother? So I was thinking about the possibility of actually using potentially fetal nucleated cells, which the baby has put into the mother's blood. But of course, the question of this sort of research is that you have to find a marker for the fetal cells. So one of the idea that I had at that time was to use the Y chromosome from a male fetus. So I persuaded a professor to let me do some experiment in the laboratory. And one of the experiments we did was actually to use in situ hybridization techniques to stain the Y chromosome. And each time you see a male cell, you actually see a black dot inside the cell. So here you can see 4 fetal cells in mother's blood. And of course, also at that time, the polymerase chain reaction, or PCR, is coming into clinical practice. So I was thinking whether it's possible to actually use the PCR to detect this Y chromosome. So we managed to do that, and then we published this paper in 1989, just the year when I qualified as a doctor. And then a few months later, the Lancet actually published an editorial called "Is It a Boy?" Because theoretically, you can actually use this technology to do the sexing of the baby. And then I qualified as a doctor and then worked 1 year as an intern. But during that year, I was constantly thinking about this field. And so I've decided after I finished my internship to actually do a full-time PhD at Oxford. And so there's a picture of myself as a PhD student in Oxford. So you see that I'm sort of covered up almost entirely. And the reason is because I'm using a Y chromosome as a marker. And of course, every cell within my body has a Y chromosome. So I'm basically trying to protect my samples from myself. But once I start working on this full time, then I realized actually the magnitude and difficulty is actually beyond what I imagined because of the rarity of the fetal cells in the mother's blood. And so after 3.5 years of working, and basically I still couldn't get a technique which will be robust enough for routine use. So my fellowship ran out, so eventually I had to graduate in 1994 in the summer. But at that time, I was fortunate enough to actually meet my wife, Alice, who is shown here in our graduation ceremony. And I went back into clinical practice and also work on this project on a part-time basis for the next few years. And then in 1997, the year when Hong Kong went back to China, and so I decided to come home with my wife, and so joined the Chinese University of Hong Kong. This is in our lovely campus in northeastern part of Hong Kong. And I remember actually about 3 months before I left Oxford for Hong Kong, I saw in this journal of Nature Medicine, there were a couple of papers which talking about cell-free cancer DNA in the blood of cancer patients. So then I thought, well, the baby living inside a mother is actually a little bit similar to a cancer growing in a cancer patient. So I started to realize that maybe for the last 8 years, my work was in the wrong direction, that I was looking in the wrong parts of blood, because in the previous 8 years, I was trying to look at the cellular fraction of blood. But I'm thinking that maybe I should focus on the cell-free fraction in the plasma. But then the problem is that at that time, I don't have a lot of resources, and I've never actually handled plasma before. So I was thinking, how can I access the DNA in the plasma? And then I remember when I was in my college in Oxford, sometimes when I had enough of the college food, I would actually go back to my room and cook instant noodles. And of course, to cook this, what you do is that you boil the water and then put the noodles in. And then a few minutes later, you have your dinner. So I was thinking, is it possible maybe we can just boil the plasma, and then maybe that boiled soup might have some fetal DNA. Now, so you say that it's very crude, but interesting, even with this crude preparation, I was able to actually detect the Y chromosome signal in maternal plasma and serum, which has been boiled. So this is the first discovery of cell-free fetal DNA in maternal plasma and serum. And it hadn't been shown before because previously people basically just focus on the cells and throw away the plasma and serum. And then we know that actually this fetal DNA is present very early on, from like the 7 weeks of pregnancy onwards. And actually by the 10 weeks of pregnancy, concentration reaches almost 15%. And then after delivery, it is actually clear extremely quickly. For example, here you can see a number of pregnant women after cesarean section, basically fetal DNA drop very quickly, and within 2 hours it's gone. So now, with those background information, we thought that we can now start to see whether this technology can be used for prenatal testing. So we start simple, and just to detect the sex of baby, because it's useful in sexing disorders like hemophilia. And we got close to 100% accuracy. And the second thing we test is to try to see whether we can use it to detect the rhesus D blood group type, which once again is close to 100% accuracy. So the sexing and fetal rhesus D genotyping are now in routine use in many parts of the world. And then we can consider more challenging applications, that the detection of trisomy 21. But of course, trisomy 21 is a quantitative diagnosis. You have to count the number of chromosomes you have inside a cell. But of course, the challenge here is that here I don't have a cell. Basically, DNA is swimming in cell-free from outside the cell. So it actually took us 10 years to do this. So initially, we're trying to think of ways in which we can try to enrich for fetal DNA, but those technologies are not robust enough. And eventually, we thought one of our challenges in this area is that the fetal DNA, which is shown in yellow here, is surrounded by great excess of maternal DNA, which is in red. So basically, when you analyze, maternal DNA is going to interfere with your analysis of fetal DNA. So we thought instead of analyzing it en bloc, how about if I separate those DNA molecules into many, many reaction tubes, so that each tube will only have either one molecule or no molecule. So in this sort of situation, basically that the fetal DNA will be isolated on itself. And so we did that, and then we managed to get to work and actually can detect Down syndrome in certain cases. So we published that in PNAS in 2007. But interestingly, within a few weeks of our publication, there's another paper on basically the same idea. And interestingly, actually, the two papers were submitted on the same day, so May the 9th, 2007. So basically, we have competition. Now, this digital PCR method works, but it's not perfect. And one reason is because you are losing a lot of information, because you basically only can capture the information within the primers of this PCR. And there's a lot of areas you do not target. So I think it's that possible to have a method which is more efficient in terms of information. So we thought, how about if I randomly sequence the DNA molecules in plasma? I can sequence millions because of massive parallel sequencing. And then I can map each of those sequences back to the chromosome from it to come from, because I know the human genome sequence. And in the bottom here, you can basically just quantify the signal from different chromosomes. Now, interesting, when I do this, actually we'll be able to show that in trisomy 21 pregnancies, the signal in green and red, basically the chromosome 21 signal is much higher than those from the euploid pregnancy. And as a matter of fact, we're able to obtain 100% diagnostic accuracy in 14 trisomy 21 pregnancy and 14 controls. And so we published that in PNAS in 2008. And once again, actually within a few weeks of this publication, there's another one from also the same group. So then I start to become curious. So who is this other group which published this paper? So I take a look at the website and actually find that the professor in charge is Professor Stephen Quick from Stanford. But interesting thing is that he's also actually graduated from Oxford with a PhD in 1994. And of course, the same year that I qualified. So I went home and told my wife about this, and she very smartly actually dig out this photo. And of course, it's myself and my wife, and this is Steve Quick. Now, so you can see basically it seems somehow fate has brought us together. Now, interestingly, a few years later, I was fortunate enough to be elected to the U.S. National Academy of Science. And then on the date when the result was announced, I saw my name. Of course, I was very happy. And then a few lines down, I actually saw Steve Quick's name. So somehow, fate brought us together again. So this is at the picture at the ceremony in which we were admitted to the National Academy. So the three of us decided to take this picture to commemorate this. So now, back to the science. So now we've shown in a small number of cases that this sequencing approach seems to work to detect trisomy 21. So the question is that to push it out in the clinical practice, we have to do a clinical trial. So which we actually finished in 2011, January. So we find that we're able to screen for trisomy 21 with an accuracy of 99.7%, which is very, very encouraging. So we like to be able to push this technology out in the clinic. But of course, being in university, I couldn't do it myself, right? So I have to collaborate with companies. And of course, working with companies, they will only work with you if you have patents. So we also learn how to patent our technology. And then, of course, there's a learning curve. So initially, our university actually licensed the technology to a company in the UK, which they actually sat on it for a number of years and didn't do anything. And eventually, they actually gave it back to us. They thought it was useless. And then I remember I went on a conference to Thailand, and then I met this gentleman who is Dr. Charles Cantor, who at that time was the chief scientific officer of Sequenom, which is a company in the US. So two of us start talking, and eventually we start to collaborate, and then Sequenom actually then licensed our technology. But in Stanford, of course, the professors have a lot of experience with commercialization. And so Steve Quick licensed technology to a company called Verinata, which was then acquired by Illumina. So basically you have then two companies, Sequenom and Illumina, having, the similar technology. And so they fought a series of intellectual property legal battles, which was fortunately eventually settled. And so both the Stanford and our patent were pooled together to form a patent pool, which people can then readily access. So the technology was launched in October 2011. And now you can basically access it in dozens of countries around the world. And annually, millions of pregnant women undergo this process. Now, for example, in China alone, every year, maybe 1/3 of the babies born actually had taken NIPT. So this is one of the most rapidly adopted genomic tests ever. But how about Hong Kong, which is the town when I was born? And so in Hong Kong, we have basically given the technology free for the highest risk pregnant women to the hospital authority so that they can basically deliver a free screening service to the citizens here. So you can see that basically the initial goal that I had when I set out to do this research as a medical student has more or less been done. But the question is, what next beyond Down syndrome? For example, I'm still not ready to be retired. So if you try to push this concept to the extreme, then you ask, well, is it possible to sequence the whole genome from a baby using this method? But this is a very tough nut to crack. The reason is because the genome is fragmented into millions of pieces in plasma. And then the baby's genome and the mother's genome mix together, and the mother's and baby's DNA are very similar. So how do you do that? So I've been thinking about this. And then I remember one day I actually went to see this movie with my wife, which is a Harry Potter movie, which is in 3D. So I remember sitting in a cinema and put on his 3D glasses and waiting for a movie to start. And then I remember that the Harry Potter sign actually fly out. And then actually my eyes was focused onto this H on the left side. So see, actually in 3D, it looks like almost a pair of chromosomes to me. So I realized at that moment that every pair of chromosomes that the baby has got, it has one copy from father and then another copy from mother. So basically what it means is that I need to have two algorithms to crack this problem. Because up to that point, I was thinking maybe I can have one formula to crack this. But now, at that movie, I thought, now I need two formulas. So basically, let's say how it works. Now imagine if this photograph is a father's genome, and this one is a mother's genome. So basically, a baby has one half from the father and the other half from the mother. And then you go into mother's blood. The mother has a lot of genomes floating around, and you're trying to decode the fetal genome. So now, the first formula, the father side. So what we do is that you look at the father's genome on the left, and then you compare with the mother's genome on the right. And they'll try to find things which is only present in the father's genome, but absent in the mother's genome, but which is easy to find, like the flower. And then you go into the mother's blood, and then you hunt for a flower. So every time you get a flower, there's a little bit of fetal genome that he has inherited from his father. And then you zip all those flowers together, then out come the father's half of the fetal genome. But how about the mother's side? The mother's side is a bit more difficult because the baby's genome is floating in this ocean of mother's genomes. So any flower that a mother gave the baby, the mother will have it herself, so you couldn't use this technique. So instead, you have to use something different. So basically what we're thinking about is that if you look at the left-hand side of the mother genomes and the right-hand side, they are present in a ratio of 1:1. But now imagine if a mother pass on the right-hand half to the baby, and the baby releases a bit of that into mother's blood. So inside mother's blood, there should be more right-hand half compared with the left-hand half. So basically what you do is that go into mother's blood and then you count the number of halves. So the half which is in excess is a half inherited by the baby. Now, so to test whether this whole concept works, we recruited a couple from our hospital, and the mother's 12 weeks pregnant, and both the father and mothers are carrier of beta thalassemia. So to show you how difficult this project is, now for Down syndrome testing, you basically sequence several million molecules, and you have a test which is over 99% accurate. But now for this genome sequencing, we have to sequence 4 billion DNA molecules, which is like covering the genome 65 times. Actually, this project, when we started, it cost us $200,000 US for this one case. But this is the result. So this is a circles plot in which the chromosome is plotted in circle. And then you can see the fetal DNA signal is in blue. Basically, the entire fetal genome is present in blood. And then actually the gene for beta thalassemia is on chromosome 11. So I'll zoom in, and then you can see the baby is a carrier for beta thalassemia. So this work was published in Science Translational Medicine in 2010 and was chosen as a cover article. And then about 18 months later, 2 groups from the US, one from Jay Shanduri group and the other one from Steve Quick's group, actually replicated this data. Now, so you can see how this field is developed, right? So I was initially looking at the fetal cells in mother's blood, but then the cell-free cancer field inspired me to look for the cell-free fetal DNA. And then the fetal side is actually going very well. So I think it's about time for us to feedback what we learn in fetal field into cancer, because cancer is a #1 killer in many parts of the world. So one cancer that we have started with is a cancer called nasopharyngeal cancer, or NPC for short, which is a cancer which affects this part of the head and neck region. And this cancer is particularly common in the southern part of China, where I live in. Now, for example, in Cantonese men, the lifetime risk of having this cancer is about 1 in 39. And another reason why we chose nasopharyngeal cancer is because for this cancer detection method to work, you will need to have a molecular target which is specifically associated with cancer, just like the Y chromosome is associated with male fetus. So for this cancer, we have that in the form of Epstein-Barr virus, EBV, because almost all cases of this cancer in China has the EBV genome inside tumor cells. So I was hypothesizing that maybe in the course of its biology, this NPC tumor cell will release this EBV DNA into the bloodstream. And I still remember when I started this project, I was asking the clinician, and actually many clinicians don't believe me because they think that since most of us are chronic carrier of EBV, so if I do a technology like that, I'm going to have a lot of false positive result. But I still remember I still decided to go ahead. And this is the result. You can see on the left-hand side, an NPC subject basically is stuffed full of EBV genomes in the blood. And on the right-hand side, then healthy individuals, most is negative. So we think that maybe this technology will have use in early detection. And this is worthwhile because like many cancers, the earlier you see NPC, the better is the prognosis. For example, you see it in stage 1, then long-term survival is over 90%. But you see it in stage 4, then it dropped to just over 60%. But despite the healthcare system in Hong Kong being rather good, still some 76% of nasopharyngeal cancer cases in Hong Kong only present in stage 3 and stage 4. So many people are dying, and we want to do something about that. So a few years ago, we proposed a project whereby we'd like to screen 20,000 subjects in Hong Kong over a course of 3 years, and like to focus on the highest risk group, which is males between age of 40 and 60, and try to see if we can actually use this plasma EBDNA to detect the cancer earlier. So every week we send a team to each of the region in Hong Kong, and every week we recruited about 200 cases. And in the end of the project, we actually recruited over 20,000 subjects, and amongst those, we identified 34 cases of nasopharyngeal cancer. Now, just to recap, so without the screening, so without this technology currently in Hong Kong, most of nasopharyngeal cancer is detected in late stage. So stage 3, which is in orange, and stage 4, which is in red. But now, with this technology, you can see that we can change the stage distribution completely, so that most of the cases detected are in stage 1 and stage 2, which is the green and blue. And also because they're so early, that actually only 1 out of the 34 cases died on follow-up. The rest basically were cured. However, if you don't have a screening in this historical unscreened cohort, then the chance of dying is much higher. It's actually 10 times higher. So this work was published in the New England Journal of Medicine in 2017 and was chosen by the journal to be one of the 10 notable articles that year. But nasopharyngeal cancer is only the beginning. So to demonstrate this technology would work, and actually by using this circulating DNA, we can basically apply to other cancer. Just like in the fetal case, I can detect the entire fetal genome there. So in 2013, we actually published a couple of articles in which we basically call this technology cancer genome scanning in plasma. So in which we actually can scan for, in this one, the genetic alterations like copy number aberration, which is in a way similar to the trisomy 21 technology. And then we can also detect single point mutations. And then in the second paper, we can basically detect the methylation changes, the epigenetic changes associated with the cancer. For example, if you use this technology in circles plot, you can see the copy number aberrations in different parts of the cancer genome in blood. And then you can see the methylation changes as well. And you can apply it across multiple types of cancer. But of course, in this cancer type of screening, if you get a positive signal, you still have to ask, where is the cancer? So what we did is that we then scan the epigenetic status of different tissues in the body, and then we basically develop a method whereby from the DNA sequencing data in plasma, in which we look at the epigenetic information, we can deduce what are the relative contributions from different tissues. So we call this technology plasma DNA tissue mapping, which is basically a way to predict the tissue origin of a cancer. And then we actually work with a company called Grail, which has actually recently launched an epigenetic multi-cancer screening test called Gallery. So basically this test is the first multi-cancer early detection test. And I expect there will probably be many other similar tests in the future. And hopefully with all this, we can basically save lives from cancer. So in summary, I think it's advantageous to research into the non-obvious. For example, when most people are talking about DNA being inside cell, we actually like to look at DNA outside cell, which led to discovery of circulating fetal DNA. And also it's interesting to be able to see similarities between apparent different phenomena. For example, we saw that similarity between the pregnancy model and also that of cancer. And also think that for this type of work, clinician scientists are very valuable because as a clinician, we're able to see what the patients need. And as a scientist, we see what the laboratory can deliver. And finally, I think it's a synergy between academic research and commercialization. Because if we just focus completely on academic research, we don't have the mechanism to push this out into clinic to benefit the patient. And so with all those, then I believe that the era of NIPT and early cancer detection is with us now. And hopefully with this, the patients is going to benefit. So finally, I'd like to thank individual from my group for generating data which I presented to you just now. And thank you very much for your time.
Pues bueno, qué mejor forma de acabar esta mañana con esta conferencia inspiracional de el que le digo yo, creo que va a ganar un premio Nobel con todos sus descubrimientos y de verdad que para nosotros fue un placer estar con ustedes estos 2 días. Sé que es un tema que no era fácil de manejar, pero creo que todos hemos salido con conceptos pensados y por lo menos con muchas ganas de estudiar y de aprender más. De parte mía, de parte de todo mi grupo, de parte de todo el grupo organizador, creo que este es otro evento exitoso de los que realizamos cada año. El próximo año esperamos hacerlo presencial, si las cosas se permiten. También queremos agradecer enormemente a todos los que han estado acá todo el tiempo y a los patrocinadores, a Voz La Francol, a Bayer, a Laboratorios Exceltis, a Cordol de Vida y Colgenes, a General Electric, a Promecto Mindray y a Genetics, quienes se han vinculado para tratar de abaratar costos y que les llegara a todos ustedes este evento de gran calidad. Yo los espero el próximo año. Nosotros estamos atentos a recibir observaciones. Si quieren algún tema en específico, siempre somos, digamos que tratamos de estar a la vanguardia. En estos 30 años de empresa siempre nos hemos reconocido por innovar y también lo queremos hacer en nuestros cursos. Así que si tienen alguna sugerencia, háganselo saber a mí o a cualquiera de los compañeros. Muchísimas gracias por toda la atención y los esperamos el próximo año. Hasta luego.